pleased to be here in this meeting to give a little talk about my last results that we got related about a new way to do proteomics for venomics. So we know that the publications related to applications of omics approach related to venoms has increased over the last 20 years, and especially the proteomics studies. And we have classical ways to do the proteomics. One of the most applied is the classical trypsin-based protocols, but we have some problems related to that. So there is the fact that digestion efficiency depending on the sequence, and also we can have some oversampling of the most abundant peptides, and we need to have uniform cleavage sites in the sequence. So there is a trend that has emerged to overcome these situations, and is the application of another enzymes other than trypsin. And in this way, we can have to increase the number of proteolytic peptides and have a better covered sequence, improving the characterization of the PTMs. In our group, recently, we developed a new methodology that is named Multi-Enzymatic Limited Digestion, or named MELD. That is an approach that we can have single step with application of several enzymes together diluted in a mix. And it's interesting that we have a limited time for this reaction, and it's a quite fast reaction of 2 hours. And in this way, you can increase the number of unique peptides and have a high confidence in the sequence validation, the de novo sequence, and the PTM characterization. So until now, we didn't have applied yet this methodology to study venoms. So after that, we question ourselves, how will MELD perform compared to trypsin digestions for inventorying the snake venom arsenal? So for that, we chose to study 2 different venoms. So we have the Dendropiasis polylepis venom, a species from Elapidae family, and the Echinocellatus venom, a species from Viperidae snake family. And we know that they are completely different venoms. So we can see here in the gel, in the 1D gel, that mamba venom presents several small molecules compounds, probably related to 3-finger toxins, protease inhibitors, and Echinocellatus presents a higher molecular mass content. And with the 2D gel, we can see that probably they are isoforms and they can be related to metalloproteinases and serinoproteinases. Also, we can see that in the database from UniProt, we have few deposits related to these snakes. So this reinforces the how it's important to us to have proteomics studies related to these venoms. So for that, we selected 10 micrograms of venom from each snake, and we proceed for sample preparation with a reduction, alkylation, and a second reduction. And we divide the groups in 2 protocols. So the first one, we proceed with MELD, which applies trypsin, glucine, and chymotrypsin. And the second group, we perform the classical digestion with trypsin. And then we analyze the digested peptides on a QS-active mass spectrometer, and we did the bioinformatic analysis using applying the PixX Plus software using a UniProt database that we created with snake plus venom keywords. So first we can see here the results for the Dendropiasis polylepis proteome, and we obtain a quite similar amount of spectra during the data acquisition for both protocols. And after we set all the parameters to do the database search, we see that from the 7 parameters that we analyze in this database search, 4 of them showed to be improved by the protocol of MELT. So this was quite interesting for us. And when we look to the venom components distribution, we see that there are quite similar distributions of toxins, non-toxins, cellular components, and protein family not assigned between the both protocols. Otherwise, MELD shows to have a small increase in the coverage of toxins and non-toxins, and less cellular components and uncharacterized proteins from UniProt. As expected from the toxins, mamba venom has a huge expression of three-finger toxins. And in this proteome, we also found a lot of cellular components and nerve growth factors, what is quite curious, and I will discuss later about this fact. But we found some groups of toxins that were described depending on the protocol that we applied. So we see here that we have MELD for, we found hyaluronidase and natriuretic peptides. Some of them were never described before in this proteome, while for trypsin, we found cathepsins and hypotensins. So for the echisocelatus proteome, we also found a quite similar data acquisition of mass spectra for the analysis in both protocols. And after we did the database search, we see that we had a huge improvement of the coverage of the sequence in this database search. So MELD showed to be a very interesting approach to be applied in this kind of viper snake type. And for toxins, you can see here that we had coverage very interesting about the toxins, and we didn't find cellular components, neither protein family not assigned for MELD protocol. And we can see that related to this proteome of echisocelatus, as expected, this venom presents several isoforms of snake venom metalloproteinase, what we found in both protocols. And we also found cellular components in NGF only with trypsin. And with trypsin, we also found nucleoxidase family and glutathione peroxidases. And for the MELD protocol, we found C-type licitine-like and VGF compounds. So some of them were never described before for this kind of proteome. So during the presentation, probably we ask ourselves why you had so much cellular components and in NGF in mamba venom. To answer that, we have a hypothesis that may be related to their fangs. So we know that Dendropiaspis polylepis is a proteoglyphide snake. So this snake has a non-fused phenotype, while Echisocelatus is a solenoglyphide snake with a phenotype, fused phenotype of the fangs. So we can see here in these pictures how the fang is characterized and leads to isolation of the venom from the saliva for the viper snakes. So probably this type of fang for mamba lead to a saliva contamination, and that's why we have so high content of cellular components in their proteomes. And about the NGF, I brought some papers here to show that NGF is produced by, in the saliva and is stimulated by the mastication and the oral food healing. So probably during the milking process, we have some stimulation of expressions of the NGF in this venom, in this saliva from mamba, and we have so high content of NGF. Summarizing all these findings, you can see that MELD presents a different coverage according to the presence of higher molecular masses contained in the venom arsenal. So it's quite interesting that this is a powerful strategy for viper venom type. We obtain also similar results comparing Naja species, that is elapid snake species, and beets, a species from Viperidae venom family. So we can see that we have validated some of these finds with another species. Also, this strategy can be applied to identify new venom components and increase the in-deep sequence of toxins. And this may represent an innovative way to do venomics and may open new perspectives for sequencing of the venom arsenal inventory. So this project is a small part of a multi-centric project named Addon Venom. If you want to know more, you can scan the QR code in the screen to follow us about the updates of this project. Also, we are on Twitter if you want to follow us. And this project is developed under the supervision of Professor Louis Quinton. And together with my colleague Damien, we are the venomics team from the MS lab at University of Liège in Belgium. And we also have a website and Twitter if you want to know more about our group and also about our updates. We are very open group and we are open also to new collaborations. So thank you for attention, and if you want, I am free to answer any questions that you may have. Thank you.